I am a professor of food chemistry, so I think a lot about the chemistry of foods. I was asked to take a look at not just foods, but over the counter medicines lately and look at the food dyes in them. So we thought this was kind of an interesting challenge. I've got a long standing interest in food dyes in foods, and so we thought this would be kind of a fun project. I'm going to give you the food scientist's perspective because I don't think we've heard enough of that. When we think about a food, if you think about this, the way that you choose a food is by appearance. So, appearance is very important when it comes to foods. Visual perception, especially in regard to color, is by far the most important factor when a consumer chooses to eat a food. If a food doesn't look appetizing, we will generally avoid it. For example, one of the examples that people like to use is brown water or black water. There was a product that came out recently. It was called Black Water. It failed almost immediately because it didn't appeal to people. Weird concept. Um, so, color is a key component of appearance, and essential in terms of human safety and the evolution. If a food doesn't look good, it was a way for us to know don't eat it. It could be potentially dangerous. It's also all the bright and vibrant colors in fruits and vegetables was a clue to us that we might want to eat those foods because that's where all the phytonutrients are, vitamins, minerals, things like that. Um, so I think Dr. Lippman might have touched on this earlier, why we use coloring agents in edible products. And again, this is to simulate the color that's perceived by the consumer as maybe being natural. And this is really critical when it comes to medications, especially medications that are designed for children. For example, a lot of these medicines are bitter. In general, most medicines are bitter. And so flavor can be used to cover up the bitterness, but flavors aren't perfect. So you might add a cherry flavor to a medicine that's bitter, but it's not going to really taste cherry to the consumer unless you put a coloring agent in there to give the visual perception of the berry flavor along with it. Um, we also use colors to offset color loss due to light, air, heat, moisture, and storage. We can use colors to mask the natural variation in foods. For example, annatto is a kind of a yellow color, natural color that's added to butter to maintain the yellow color of butter because butter changes throughout the season because cows are either fed hay or they might be out in pastures. There's more carotenoids in the grass. And so during the winter, there's less carotenoids in their diets. Carotenoids are these big lipophilic molecules with a yellow color associated with them, and they end up in the butter. And so butter naturally changes in colors. So for some reason, consumers like their products to look the same all the time. So we can enhance naturally occurring colors. I think it was pointed out that we can provide brand identity to products. This is extremely important when it comes to medications, and I think Dr. Taylor pointed that out. That this is a visual clue to help people recognize what pills to take and what pills maybe not to take. Um, we also use it as a decorative or artistic purposes, such as in cake icings and colors of foods. And this is generally to create visual appeal. I almost put a picture in of our secretaries had a cookie decorating party this last week, and the cookies had every single color you could think of. They were just awful looking to me, but they had a great time, and so that's a great staff-student bonding experience. Um, and we also can use colors then to attract certain consumers. Colors are often used in foods to attract certain consumer groups, like children, for example, green ketchup, which was another product that failed remarkably. So what is the basic chemistry of some of these coloring agents? I think this was pointed out earlier. Um, Most coloring agents will have a ring structure associated with them, a benzene ring. This can be one benzene ring or it can be several rings fused together. They also will have, I'm not sure how this Can you see that? Yes, no, no. They also can have extensive conjugated systems. So what that means is they've got double bonds separated by a single bond next to a double bond. It's that structure that gives them the color. That's called a chromophore. So these extensive conjugated systems is what makes a molecule colorful. I think this was pointed out earlier, they're typically large hydrophobic molecules, but we can stick groups on there like sulfate groups to increase the water solubility of these compounds. And for example, this is an example of, here you've got an azo functionality, two nitrogens bonded by a double bond, and that is a colorless compound, but if you add these benzene rings, you'll get an orange color, and that's because you increase the conjugation in that system. This has been pointed out by several of the earlier speakers. We have two different types of classes of coloring agents. We've got certified colorers, and these are synthetic compounds that are used to create these intense uniform colors in foods, drugs, cosmetics, and also medical devices. And this is because they can blend easily. And they can be mixed to create a variety of different hues. We've talked about the fact that each batch has to be certified, and that the FDA has a branch associated with it that certifies the dyes, and these specifications are listed under Title 21 and Part 74 and 82. We also have compounds that are exempt from certification. These are your natural dyes. These are pigments and dyes from natural sources such as vegetables, minerals, or animals. And these specifications are also listed in Title 21. And this is just a table of the food coloring agents that are subject to certification or exempt from certification. Again, you've got the azo dyes, phenylmethane dyes, xanthine dyes, and sulfonated indigo. These all have to be batch certified. And then you have compounds that are exempt from certification, and this includes everything from grape skin extracts to titanium dioxide to silver, gold, whatever. I bet you didn't know those were in your food. So why don't we all just use natural sources of colors in foods, right? If we are worried at all about synthetic dyes, why don't we just use natural ones? And I'm going to tell you from a food scientist perspective, natural food dyes are not necessarily safe, and they're very difficult to deal with. They are a very diverse group of molecules. There's hundreds of various naturally derived pigments. They're chemically very different from each other. Many are present as multiple derivatives, so they've got other things attached to them, different sugars, acyl groups, different things that change the chemistry. They are cultivar specific, and the levels also will change throughout the growing season. They also, unlike synthetic food dyes, can have flavor associated with them, and you may have to use them in very high concentrations because they're not as vivid as some of these other synthetic food dyes. So if you have to use a lot of them, and they also have flavor associated with them, they may not be usable in a food product. There are actually, a lot of them are fairly bitter and not very good tasting. Often what's used is extracts, not purified pigments themselves. And these extracts, when you make an extract, you're bringing other things along with it. A good example is solanine is a toxic compound often found in potatoes. And I was given an example with a certain food company that was extracting the potato for the anthocyanins, but it also co-extracted the solanine, which was toxic. So you have to always be careful about what you're using and how you're using it. The other problem with natural compounds is they're chemically active, they're sensitive to light, to oxidation, metal ions, they go reactions with each other and other food matrix components, and so they're very difficult to deal with when you're formulating and manufacturing foods. These are the FD&C coloring agents, and a picture actually of the color of each one of these dyes. This is right out of our lab. I can't remember what the concentration of these are, but you can see we've seen these structures before. We have the azo dyes, which are the red number 40, and this is hard to see up here, the tartrazine, and yellow number 6. Okay, well, let's see if I can go back. All right, you can see this, each one of those have the azo function. Then you've got the triphenylmethane dyes, and you can see the triphenyl functionality. And then we also have xanthine dyes. I think somebody mentioned that this is typically the base structure that's used for a lot of the fluorescent dyes, and then the sulfonated indigo dyes. And again, a lot of these dyes have the similarity of having sulfate groups attached to them to make them more water soluble. We also have D&C coloring agents. Now, in most of the talks that have been presented today, we've only talked about FD&C coloring agents, but I'm dealing with over-the-counter medications, and so there's D&C dyes in those medications as well. We didn't measure those because we were charged with just measuring FD&C dyes, but I want you to be aware that they are in the over-the-counter medicines that we did look at. They're also in cosmetics, things like lipstick. You can also find them in things like mouthwash and stuff like that. Okay, we talked about the fact that these have to be certified, and I just wanted to point out that there's a difference between the dyes and the lakes. A dye is these are the water-soluble forms of the coloring agents that we're using. Food manufacturers can buy these in powdered, granular, liquid forms. They're used in a lot of different ways in the food industry, things like aqueous beverages or dry mixes, dairy products, things like that. A lake is an alumina oxide that has been coated with a dye. So that it's no longer acting as a dye, it's acting as a pigment, and it's coloring through dispersion. And lakes are useful to the food industry because unlike a dye, you can use a lake in a oil-based food system. So they're soluble or they're dispersible in oil systems. So it gives you a way to color a food that is hydrophobic. I just thought this was interesting. I looked at the batch certification for the food dyes and the food lakes, and if you look at the food dyes, okay, so we call the dyes the straights. There was over 15 million pounds certified in 2018, and another 6,000, almost 6 million, pounds of food lakes that were certified. As Dr. Duell pointed out, these are what were certified in the U.S., not necessarily all of this ended up in food, and these can also be sold outside of the U.S. as well. But it's a way to understand, I guess, how much of this we are certifying and how much is out there. So the research objectives for what we were charged to do was to measure the amount of FD&C dyes in a range of prenatal vitamins, children's pain relievers, and also children's cough, cold, and allergy over-the-counter medicines. So today what I'm going to present is some of the data on the vitamin gummies, the pain reliever syrups, and the cough, cold, and allergy syrup. And I'm just going to ask that this data doesn't get any further from this talk because I have a graduate student that would like to publish this data. So this is just for today's talk. It's preliminary data, and we'd like to finish getting her published before it gets out there. So in terms of how we did the sampling is always the biggest source of error in any analytical measurement that's made. So we thought about this, and we went out and we bought commercial products, and we bought them in California, Georgia, and Indiana. We looked at between 3 and 5 brands for each category that we looked at. 3 different samples of the same brand were analyzed independently, and we identified these by their lot numbers. In the case of the gummy vitamins, we forgot to take into account that they come in different colors in the jar. So we analyzed each color of the vitamin individually. So we have a good replication of our samples, and I'll show you why this is really important. Our extraction methods were based on the method of Yang and Zhang. We modified these so that we could get a quicker method. We now have an HPLC method that's about 17 minutes long, and it separates all of the colors. We use what's called solid phase extraction to help pull the dyes out of the food products, and we use an Oasis wax column to do that. And the way this works is that the solid phase extraction matrix will bind with the food dye through weak anion exchange interactions and also hydrophobic bonding interactions. And then once we separate it from the food matrix, we can then elute it or remove it from the column and analyze it. One of the things that we are finding in this is that the food matrix plays a big role in how much you recover out of the different foods. So you may need to modify your extraction techniques to match the food matrix, and so you've got to validate your method for each one of the different matrices. Looking at this, these are just some chromatograms. I hope you can see them. I can't see any. You can't see. It's like I'm looking at a blank screen hoping I'm saying the right thing here. Anyways, what we have here is a chart of the different dyes, and these are the standard curves. This is the HPLC chromatogram, so that's a picture of the dyes separated on the analytical instrument. And then we have the limit of detection for each one of the dyes, the linear dynamic range for the assay, and then how much recovery we got. So these are just things that chemists like to see. The rest of the world doesn't really care. So we looked at, the first category we looked at was children's multivitamin gummies. We looked again at 3 brands. We had 3 independent lots of the brand. This is why we had to look in different states at times. We had brand 1, and in brand 1 we had 2 different colored gummies. We had an orange gummy, or 3 colored gummies, orange, red, and purple. The orange gummy actually was colored with annatto, which may get to your question, the gentleman in the corner, were about the different dyes. We did see different dyes in there. And then brand 2, we had orange, red, and purple gummies, and brand 3, we had yellow, red, and green gummies. And these are the levels of the dyes in the gummies. You can see in the purple gummies, we had an average of 72.56. This is milligrams per kilogram. To make purple, you need red and blue, so we also had brilliant blue in there, and we had 3.23 milligrams in there. And then in the red gummy, we had 27.06 milligrams per mil. Now, when I look at this data, the first thing that jumped out to me was that the levels of the dye seemed to change with the lot number of the vitamins that we were looking at. And so there was significant lot variation that was seen, and it seems to be related to the expiration date, which tells me that in the gummies, the dye is not stable and that it's potentially breaking down. So when we're doing assessment studies, we need to take this into consideration. Also, if you look here, you can see the relative standard deviation for these measurements is relatively high, showing that there is a lot of variability between these lots. Now, as a food scientist, I'm like, yeah, of course there is. But these are over-the-counter medicines, so I was a little bit surprised. I thought that this would be a little bit smaller. So we wanted to take a bit of a closer look at this. And these are pictures of the vitamins. This was the lot that expired 9/19. We had another one expiring 6/20 and one expiring 9/20. And you can see yourself that the vitamins, I think you can see yourself, the color changes over time because the dyes are not stable in that gummy matrix. The orange dye, which is the one at the top, you could see annatto. This is a really good example of why a natural food colorant is not always the best choice. It breaks down and it looks pretty awful with time. So what we found was that the vitamins with the oldest expiration dates had the less color, and that this correlated to the amounts of dye that we were measuring in each one of those vitamins. So I'm not sure what that says about the vitamin C in those gummies. If the dye isn't stable in the gummy and vitamin C is much more labile, what is the level of vitamin C in those gummies? Okay, so Um, this color variation can be due to a lot of different things, and I just want to point out one of the difficulties with manufactured foods and compounds like this is once you manufacture, you could have very rigorous manufacturing, good manufacturing techniques, but once it leaves your processing plant or your manufacturing plant, anything can happen. Okay, I had a great picture of a Target truck in a ravine with people trying to unload stuff out of the truck. But you could see a lot of different things can happen. So we have no control over handling, storage conditions. Lights in the store can degrade the color. So where the bottle sits on the shelf, if it's a clear bottle, the vitamins sitting in the front may have less just because they've been exposed to more light than the vitamins in the back of the shelf. So there's a lot of things to consider. When we look at the level of the dyes in the second brand of gummies that we looked at, well, again, we found that the average levels for the orange dyes, in this case we had Sunset Yellow, it was about 16, and then FD&C was about 36. When we looked at the purple ones, we had a combination of both red and blue, that was around 61 and 3 milligram per kilogram. Again, we saw a big brand variation, and the vitamins with the older expiration dates again had less color associated with them. One of the other things we noticed was that some of the vitamins in the bottle looked green to us, but they had the same shape as all the other purple vitamins in the bottle. And so we thought, is this a different color? We only found it in one of the bottles that we looked at. So we weren't sure if something happened to these or if it was truly a different color. So we measured these independent of the purple values, and what we saw was that the levels of the FD&C red were much lower, and the FD&C blue was about the same. So was this a mistake? Was this purposeful? I have no idea. It does look like a different color profile to me, so maybe somebody was trying to make green gummies on that one day in that one processing plant. It can happen. Then we also looked at the third brand, and in this case we found much higher levels of the red dye in the red vitamins. In the yellow vitamins, we found a little touch of blue, which is not unusual, fairly high levels of the yellow number 5, and then the brilliant blue was around 11.5 milligram per kilogram. Again, this is the yellow vitamins out of that jar, and I think you can see yourself that there's a huge amount of color variability in that jar. So we wanted to understand the variability. We analyzed 6 different vitamins that were of this yellow color, kind of yellow-green color, and we found that the average FD&C yellow levels were about 28, and blue were around 1.6. When you make these measurements, what's really important to always notice is the relative standard deviation, because that tells you how good that number is. And so we're seeing a lot of variability within the jar, between the jars, and then within the color of the vitamin itself. And this is just kind of a summary of the vitamin data. We then took the data that we measured and we calculated it based on milligrams per gummy. And you could see that, yes, we've got variability in relationship to expiration dates and different lies different levels due to different dye lots. But in general, the levels were very low in terms of milligrams per gummy. All of them were less than 0.4 milligrams per kilogram per gummy. So we thought that was pretty good. By far, the red was the highest level. When we looked at the dyes in the pain reliever syrups, we saw a similar kind of thing. When we looked at one brand, we found levels that ranged around 8 for the red, number 40. Red was always present in all of these samples, and then blue in some of the samples. We didn't see green in any of the samples, which is very similar to what Dr. Duell found as well. So you could see that it ranges again. We looked at 3 different brands. One of the things to note when we looked at these brands, You might see that FD&C 40 was on the label, and we were able to measure that, but we also saw that there were D&C dyes in there. And again, we didn't measure the D&C dyes for these. So when you look at the cough and cold and allergy syrup, we saw the same thing. Less variability in these because you're looking at homogeneous samples. They're liquid samples. And so we went through and we looked at 5 different brands there. Again, in some brands we have the FD&C, and in some brands we have a mixture of the D&C and the FD&C dyes. And in summary, what we found was that the pain relievers, when we looked at the amount milligrams per mil pain reliever, these were significant, and they ranged between 0.64, this is for red, number 40, and 10 milligrams per mil. And then with the blue, they ranged between 0.03 and 1.45 milligrams per mil. And remember, when you're taking a dose of this, you're taking multiple mils, 5 to 10 mils, 5 times a day. So this turns out that this was actually a significant source of dyes, especially if you're considering a child that needs to take an allergy medicine every day over allergy season. So, and I think Dr. Brenman will talk more about that in a few minutes. So in conclusion, the dyes that were present in the children's gummies and vitamins included the FD&C 40, the FD&C number 5, and relatively low levels of the FD&C blue. And in one case, we had FD&C 6, that's the sunset yellow. The levels of dyes in the gummies didn't exceed 0.3 milligram per gummy for all colors, so we thought that was pretty good. The primary FD&C dyes that were present in the children's pain reliever syrups and the cough, cold, and allergy syrups included the red, number 40, and blue, number 1. We also found D&C colors present in those medications, but again, those weren't measured. And what we found was that the levels of the dye in some of the products indicate that the over-the-counter medications can be a more significant source of dye when dosing is considered. And again, that was just the ranges that I showed. I just want to acknowledge Arlie Lemkuller, who is the graduate student that did all the analytical work on this, along with our collaborators in the lab, and then Dr. Mark Miller and Asa Bradman and Rosemary Kastoriev, because they've all helped in one way or another with putting this together. So thank you.